Okay, thank you, Barbara, for this opportunity. I'm sure everyone appreciates the work that you put on organizing this, along with Dennis and along with the people uh, England. So I'll try to start my presentation right now. So I'm going to talk a little bit about some research that my lab has worked on, and that is based on something that I've made during my master's and my doctorate, and I'll share with you guys some of the results that are coming right now. Okay, I'll try to let the data speak for itself, and I'll try to uh, highlight the work of the students, which is way more important than my work. Okay, so we'll talk a little bit about zebrafish defensive behavior. Then we will turn to a little bit about glutamate and nitric oxide, which is something that started, chronologically speaking, after I started working with serotonin, but I think it's going to tie a little better in the narrative that I'm trying to construct here. Then we will talk a little bit about serotonin, which is my favorite neurotransmitter because it's so complex and it has so many layers of complexity. And then a little bit about interactions between glutamate, nitric oxide, serotonin, and put all of this into a mechanistic framework, okay? As I was saying, I'm referring here to zebrafish defensive behavior instead of speaking about anxiety and fear, because this is a highly disputed definition in the area. Neuroscientists, behavioral neuroscientists, are discussing whether or not this subjective states, emotional states, which are called anxiety and fear, can actually exist, or if they exist, can be studied in animals, because, as I've said, they are subjective. However, there are core components which are behavioral and physiological components which are part of this defensive behavior that can be used to model, or at least we hope they can be used to model these things that we call fear and anxiety, which can be relevant to understanding anxiety and fear disorders, okay? So in order for us to understand defensive behavior, we need to first understand that there are some threat detection mechanisms. There are mechanisms in the brain of the animal which are useful for detecting whether a threat is present or not, how far the threat is from the animal, and which are the available options from the point of view of defensive behavior, okay? But what is more important for us here is not the threat detection mechanisms per se. What I think is more interesting is that response selection mechanism. So if a threat is present or not, if a danger is present or not, and if it's far away from the animal or if it's near the animal needs to respond accordingly. If the animal doesn't respond accordingly, doesn't behave accordingly, the animal gets eaten, the animal can die, okay? So how is that response selected? There's a theory from the 1970s, which is called defensive distance, or the predatory imminence continuum, which will state that there are at least two characteristics of that drive response selection mechanisms. One of them is defensive distance, which is a psychological construct. It's not a real distance per se. It's not distance in meters, it's not a distance in centimeters, but it's a psychological distance, whether the predator is present or not, whether the danger is potential or if it's real, and if it's real, whether the danger is distal or proximal, okay? And the second element is whether there are escape routes available, for example. So in this figure here, this is shown. So if there are escape routes, if flight is available, the animal behaves when danger is present, but it is distal by trying to escape. If the danger is proximal, the animal threatens the danger and the animal attacks the danger, okay? If the flight is not available, the animal behaves differently at distal defensive distances, distal threats, distal dangers, by freezing to try to avoid detection instead of trying to flee. And as the distance increases at higher distances, the presence of the threat, for example, a predator, is uncertain, okay? So the animal tries to change its movement patterns in the environment by entering a state of careful exploration, which we would call an anxiety-like state. Okay, so there are many behavioral paradigms which have been proposed for zebrafish, okay, for using, for testing behavior in these different defensive distances, okay? So, for example, this appeared in some other lectures as well, and other presentations as well. For higher distances in which threat and danger is uncertain, we use the light-dark test, the novel tank test, open field test. That is, all types of tests in which there is conflict and which there is no certain threat, okay? When the threat is distal and behavior turns into a flight or freeze mode, we use anti-predator behavior by exposing the animal to predators, for example, or we can use the alarm substance that is produced by some skin cells in the animal, by club cells from zebrafish, and alert animals around here, around the focal fish, if there is some threat because the animal has been injured, okay? So these are the kind of models that can be used to study zebrafish defensive behavior. Of course, there are lots of other ways of studying it. Those are the most common and the most advantageous so far to use in laboratories. Okay, so turning now to glutamate and nitric oxide. So glutamate is the main excitatory neurotransmitter in the central nervous system. So it's associated with almost all major brain functions. That is a problem, and that is something interesting. That is a problem because pharmacologically speaking, if you manipulate glutamate, mainly you're manipulating a lot of other functions which are not very specific, okay? But it's interesting because it's involving so many things that there are a lot of open questions as well. So glutamate in all vertebrates so far have two types of receptors, which are ionotropic receptors, including NMDA, AMPA, delta receptors, and kainate, and also metabotropic glutamate receptors. So this is a very complex system. And what we did in our lab was focus mainly on the NMDA receptor. So at least since 2011, there is some work from Gerlai's lab showing that blocking NMDA receptors produce an anxiolytic-like effect in zebrafish. In that specific case, in the novel tank test. So if you treat the animal with MK801, which is an NMDA receptor blocker or antagonist, the animal spends less time in the bottom of the tank. It displays less bottom dwelling, which is a way to say that the animal is not very anxious. So in 2014, we did the same, this time using the light-dark test, and we did a conceptual replication of Gerlai's work, showing that treatment with this drug decreases scototaxis, which is the preference for the dark compartment, and also it decreases thigmotaxis in this animal. So this is something that suggests that NMDA receptors are involved in some way with anxiety-like behavior in zebrafish. Okay. What we also found, however, is that the NMDA receptor, this effect of the NMDA receptor, this role of the NMDA receptor appears to be fully involved in the modulation of the nutrinergic system. As some of you may know, NMDA receptors are linked physically to one enzyme, nitric oxide synthase, that is involved in the production of nitric oxide. Okay, so what we did, we injected the animal with a low dose of NMDA. We had a lot of trouble finding the right dose, by the way, because higher doses, slightly higher doses, induce an epileptic-like pattern in the animal. Okay, and what we observed is that when we injected a low dose of NMDA, this animal showed a high increase in anxiety-like behavior. So the opposite effect of blocking the receptor. What was interesting is that when we treated this animal with a drug that blocked the production of nitric oxide, we abolished the effect of NMDA. So if we activate the receptor, the NMDA glutamate receptor, we induce anxiety-like behavior. If we block nitric oxide synthesis after activating this receptor, we block the effect of the NMDA. So this is kind of like a cascade. What we also find, and this is 1 year later, is that the NMDA receptor, and more specifically nitric oxide, is also involved in long-term sensitization of fear and anxiety-like behavior. So this is how we do this. This is called time-dependent sensitization, and we propose it as a model for post-traumatic stress disorder in zebrafish. We expose the animal to alarm substance, which induces fear-like behavior, and leave the animal stress-free, that is not exposed to the substance, for 24 hours, okay? 24 hours after exposure, the animal shows increased anxiety-like behavior in the light-dark test, okay? So we think that the behavior is sensitized after this full 24-hour incubation period. When we block the nitric oxide synthesis with L-name, which is a non-specific blocker, the same that we used in the previous work, what we observe is that if we block 30 minutes after exposure, that is when this response is incubating, the animal is no longer exposed to the alarm substance here, but when this fear or anxiety-like sensitization is incubating, what we observed is that blocking nitric oxide blocked this time-dependent sensitization. The same did not happen when we injected animals 30 minutes before exposition to the alarm substance, suggesting that it's not, that nitric oxide is not acting on threat detection, on the specific mechanisms of inducing fear during exposure, but on the mechanisms of sensitization itself. So we think that it might be involved in plasticity mechanisms. We began studying serotonin while I was still in my PhD. So this is work that is ongoing, actually. So it's been like 7 years at least that we are studying this neurotransmitter. It's a very complex neurotransmitter. There are many different types of serotonin receptors. The serotonergic system of zebrafish is more complex than the serotonergic system of rodents, for example. So it makes for very interesting questions to be asked. So back in 2013, what I've showed is that exposing the animal to the light-dark test increased extracellular levels of serotonin in the brains of these animals. And exposing the animals to the light-dark test increased tissue content in the hindbrain and in the forebrain, while exposing the animal to the novel tank test increased serotonin content in the midbrain. So we had an effect of exposing the animal to the apparatuses, which induced what we thought was anxiety-like behavior. And this exposure produced different effects on the serotonergic system. That was kind of puzzling, because at first we thought that if we're talking about the novel tank test and if we're talking about the light-dark test, we're talking about tests that measure the same thing. They would measure an underlying construct of anxiety. But if they produce different mechanisms of, if they produce different effects, for example, on the serotonergic system, they are probably not measuring the same thing. They're probably not inducing the same behavioral state into the animal. Next, we treated these animals with drugs that affect the serotonergic system. So first we treated the animals with acute fluoxetine, which increases the serotonin levels in the brain. Okay, what we observed is that fluoxetine decreased defensive behavior in the novel tank test and increased defensive behavior in the light-dark test. We could say it like that. It decreased anxiety-like behavior in the novel tank test, but it increased it in the light-dark test. That is a paradox. The same drug producing opposite results in different tests. So we thought, well, maybe this has something to do with fluoxetine itself. Maybe it has something to do with serotonin, because we already saw that serotonin levels were differently modulated by exposure to these tests. So we use PCPA, which is a drug that blocks serotonin synthesis. So when we injected PCPA in these animals, our treatment reduced serotonin levels to about 10%. Okay, so it dramatically reduced serotonin levels in the brain. And what we observed is that PCPA has an opposite effect in these tests as well, and an effect that is the opposite of fluoxetine. So if we increase serotonin using fluoxetine, we have a decrease of behavior, of defensive behavior in the novel tank test, and an increase in defensive behavior in the light-dark test. If we decrease serotonin with PCPA, we have the opposite effects. Next, we sought to understand which receptors were involved. So we used two antagonists, okay? This first antagonist, WEI-100-635, blocks serotonin 1A receptors. And the second drug, which is SB224-289, blocks serotonin 1B receptor. Okay, so what we found is that blocking serotonin 1A receptor produced the same effect in both the novel tank test and the light-dark test. So if there's an opposing role of serotonin in behavior in both tests, this is not due to serotonin 1A receptor. However, when we treated the animals with the serotonin 1B receptor, we had some hints that it decreased defensive behavior in the novel tank test, but it appears to increase defensive behavior in the light-dark test. So if there's an opposing role of serotonin in both tests, it probably is related to the serotonin 1B receptor. We also sought to look into interactions between glutamate and serotonin. So what we did here is increase serotonin levels. This is a paper that was published in 2014. We tried to increase serotonin levels, this time not by using fluoxetine, because fluoxetine can have lots of other effects. So what we did here was give 5-HTP, 5-hydroxytryptophan, which is a precursor to serotonin. Okay, so what we observed is that when we give a serotonin precursor, increasing serotonin in the brain of the animal, we produce increased anxiety-like behavior in the light-dark test. So again, in this test, serotonin is, we could say that it increases defensive behavior. When we treated these animals, we co-treated, actually it was a pre-treatment, okay? When we pre-treated these animals with the NMDA receptor blocker MK801 and treated these animals with 5-HTP, the antagonist blocked the effects of increasing serotonin. So the effects of serotonin are mediated somehow by glutamate, by the NMDA receptor. And when we pre-treated these animals with NMDA, we potentiated the effects of increasing serotonin. So they are acting synergistically. Okay, so glutamate and serotonin in the specific test act synergistically to increase anxiety-like behavior. To our surprise, when we pre-treated these animals with nitric oxide synthesis blocker, which we have already shown that is related to the effects of NMDA, we were able to also block the effects of 5-HTP. So nitric oxide also mediates the effects of serotonin. What we thought was happening was that serotonin and glutamate were acting at the same regions to induce the production of nitric oxide. And the production of nitric oxide was the next step in increasing anxiety. But this is, of course, not really easy to demonstrate. This is actually a very complex mechanism. So we have already shown that the serotonin 1A receptor is involved in anxiety-like behavior in this test. So we treated these animals with an antagonist for this receptor, and when we pre-treated these animals with the nitric acid synthesis blocker, we inhibited the effect, the anxiolytic-like effect of this antagonist. So this is interesting because blocking the nitric oxide system can either turn on or turn off an anxiety-like, anxiogenic or anxiolytic behavior. So we thought of nitric oxide as a kind of switch, of molecular switch for increasing or decreasing anxiety. And we also observed the same with the serotonin 1B receptor, suggesting that nitric oxide is downstream of serotonin in controlling anxiety-like behavior. So we had this strange effect in 2013 in which serotonin appears to be anxiolytic or anxiogenic depending on the test. So we've tried to look a little better into that. Instead of using the novel tank test or purely the light-dark test, we sought to use a stronger fear-inducing stimulus because we thought that the novel tank test was actually modeling not anxiety per se, but some kind of fear-like behavior, okay? So what we observed, what we did was give alarm substance to these animals, okay? And in retrospect, I think that we should have made a different experimental design. We followed experimental designs that were common at the time, in which we exposed animals to the alarm substance for 6 minutes. And after exposure, we put the animals in the light-dark test. Okay, so this is not supposed to be behavior that is elicited by a distal threat itself, but it's kind of like a residual behavior. It's kind of like the animal is returning to normal. He smells danger in the water. When that smell disperses, it's not very adaptive for the animal to immediately return to his normal behavior. So he gets a little bit anxious. He gets cautious because he doesn't know if the danger has actually dispelled. So we observe this residual effect in which alarm substance increased anxiety-like behavior after exposure. So when we treated these animals with fluoxetine, with a low dose of fluoxetine, we blocked the effect of the alarm substance. Interesting, if we treated these animals with fluoxetine without exposure to alarm substance, the effect was opposite. So it decreased anxiety-like behavior. Okay? Actually, sorry, it increased anxiety-like behavior. So fluoxetine without alarm substance increases anxiety-like behavior. Fluoxetine with alarm substance decreases this residual anxiety-like behavior. When we tested the serotonin 1A antagonist, however, it had no effect on this. So, but we knew that the serotonin 1A receptor was involved in fear responses, or at least some fear responses in other animals, more specifically in rodents. So we sought other assays as well. So in the second assay, what we used was try to test pain or nociception or nociceptive behavior in these animals. So we injected acetic acid in the tail of the animal, and what we observe when we inject acid is that the animal starts to beat its tail, kind of like trying to get the acid out, because it's producing pain in the animal. And what we observed is that when we give alarm substance, when we expose the animal to alarm substance, the animal no longer produces this kind of behavior. It makes sense. This is called fear-induced analgesia. Okay, so the animal stops feeling pain because at that time it has something more important to attend to, and more specifically, the fact that there might be, for example, a predator around him. So fear decreases pain. And when we treated the animal with fluoxetine did nothing with that. However, when we treated the animal with WEI-165, which is the serotonin 1A receptor antagonist, it partially blocked this effect. Okay, so the serotonin 1A receptor is involved in some aspects of fear in this animal, but it's involved specifically in fear-induced analgesia. Okay, we also observed some interesting effects of fluoxetine specifically in physiological responses. What we observed is that this drug blocked the effects of the fear-inducing alarm substance in serotonin efflux in the brain. What we observed was that when we expose the animal to the alarm substance, there's an increase in serotonin efflux. So serotonin is released, okay? Or transport stops, okay? We also observe some signs of sympathetic activation. So for example, when we expose the animal to alarm substance, we observe increased blood glucose, we observe increased hemoglobin in the blood, and we also observe increased catecholamines in the brain. Well, as I've said, there are some problems with our experimental design in this 2014 work. So, because we expose the animal to alarm substance, and after exposure, when the substance is no longer present, we observe the behavior of the animal. This was very common by the time, but then we started to see some contradictory results in the literature and in our own work in the laboratory. Because, for example, there are some behavioral effects which are observed in this kind of design. Expose the animal to substance and then observe the behavior in a substance-free environment. And in other designs in which we observe the animal during the alarm substance exposure, okay? This is important because if the substance is present in nature, the threat is probably present as well. So it's a situation of distal threat or distal danger. While after alarm substance exposure, this is just a potential threat, okay? Speaking in terms of fear and anxiety, during the exposure to alarm substance, we would have fear. And after the exposure to alarm substance, we have something that is more like anxiety. So we treated these animals with different drugs. We treated the animals with clonazepam, which is a very strong benzodiazepine that is used, for example, to treat panic disorder. Was our positive control. What we observed is that this drug decreased the behavior both during exposure and after exposure. However, when we treated the animals with fluoxetine decreased responses during exposure, and it increased responses after exposure. And when we shut down the serotonergic system, either by blocking receptors with meterglin or by blocking synthesis with PCPA, we had no effect during exposure, okay? So this suggests to us that there is not a tonic inhibition of fear-like behavior in the alarm reaction paradigm in zebrafish, but it did decrease defensive behavior after exposure. Moreover, we also observed something that we observed in 2014, that alarm substance had some effects on the serotonergic system. We don't know right now what is going on in the serotonergic system during exposure to alarm substance. My bet is that the serotonergic system is silent during exposure. However, when threat is no longer present, what we observed is a decreased activity of monoamine oxidase, which is an enzyme that metabolizes serotonin, and a decrease in serotonin uptake. Both effects would amount to an increase in serotonin availability for neurotransmission, okay? So what we think is going on is that after exposure, there's a surge of serotonin release, okay? This work is in press. It's probably, the final version is probably going to go out next month or something. It's going to be published in the Journal of Neurochemistry. And we are also very proud because this is our first work which relies completely on open science. We shared our data, we shared the materials, the work was pre-registered. So this is something that we hope gains traction in the field of behavioral neuroscience, but also in the field of zebrafish behavioral neuroscience, because it's very, very important to increase transparency and reproducibility of these results. What we think is going on, or what we think is our main contribution to the field of behavioral neuroscience, specifically to the field of zebrafish behavioral neuroscience, but more to the broader field of behavioral neuroscience, is an extension of a hypothesis that appeared first in the 1970s. That is the dual role hypothesis. This hypothesis goes like this: serotonin increases anxiety-like behavior by acting on forebrain, and it decreases fear-like behavior by acting on the midbrain. Okay, what we thought here is that it is not exactly decreasing fear-like behavior. Serotonin acts like a switch. So when threat is no longer present, so there was important signs of threat, specifically of distal threat, which would induce fear-like behavior in the animal, or fearful sensations in humans. When threat is no longer present, this surge of serotonin would switch off the mesencephalic circuits, which are involved in erratic swimming and in fear-like behavior, okay? So because fear-like behavior is no longer adaptive in this context. So if you don't know whether there's a threat or not, swimming like crazy is not very adaptive, because you might call attention to yourself, okay? At the same time, the prosencephalic, the forebrain circuits are activated by serotonin. So it switches off the midbrain circuits, and it switches on the forebrain circuits, which will lead to freezing and more specifically to risk assessment, okay? So the animal stays put and scans its environment, looking for signs of that the danger has actually gone and it's not present anymore, and it's safe to resume normal behavior, okay? So what we would first say, oh, so it increases anxiety and it decreases fear. It's not exactly like that. It's a little bit more refined, you know? It's a very adaptive function. However, of course, it also can be hijacked, that's our hypothesis, in malfunctioning circuits, which lead to anxiety. This work is part of the work of these students, okay? So Mariana and Gabriela are undergrad students. Haira is a master's student, and they have all they are responsible for this wonderful finding that we had, okay? I would like to thank you all for paying attention to what I had to say. I would like to thank you all for having patience as well with my bad English. This is my team. This is a huge team of mostly undergraduate students. One of the difficulties of making science in the Amazon is that we can barely rely, for example, on master's or PhD students. So most of the work is made by undergrad students, and they have to work like 3 times more than any other undergrad student because they have to compare with master students in other regions of the country, okay? This is a very diverse team as well, okay? I would like to thank you guys. If you were interested, you can check out our social networks. So we have Facebook, 2 Twitter accounts, one in Portuguese, one in English. We also have Instagram, so follow us there and get clicks and likes and whatnot. And this research is specifically the last point of the research that we just that I presented on alarm substance and serotonin was supported by Conselho Nacional de Pesquisa, which is the Brazilian most important funding institution. It's a public institution, and without which we would not be able to do this. Okay, so defend our institutions for funding because they are being attacked and destroyed, and this type of research would not be possible and will not be possible in the future if funding gets destroyed and taken away as it's being taken in Brazil right now. So thank you very much.